Hey scholars, welcome to chapter 17.4, looking at how can we evaluate and deal with chemical hazards. And so yesterday or whatever, the last previous session, we looked at 17.3, looking at what are the chemical hazards. Today we're going to develop and look at how can we deal with those hazards. Let's take a look at the first part. So one of the factors that determines, we're going to look at some of the, there's many factors that determine how harmful a substance is, especially a chemical. And the way that we look at that is called toxicology. Maybe you've heard of this before on CSI when they run a toxicology report, when they look at what was the person overdosing and that's how they died. And so toxicology is kind of the study of the chemical makeup inside of a person, and you can do that while they're living or like in CSI mostly when they're dead. Toxicity though depends on a number of different things. The first one is the dosage, and you guys have already kind of looked at that, or maybe you will look at that as far as the lab goes with the crickets and looking at dosage. It also has to do with the age of the person or the organism, as well as their genetic makeup. And some organisms have what we call MCS, which is multiple chemical sensitivity. So they can be sensitive to multiple chemicals at the same time. If those combination of chemicals combine, they can produce some sort of reaction as well inside the organism. Also, it has to do with the solubility and persistence of the chemical that you're testing for, and that also can lead to biomagnification. Like, for instance, in mercury, as mercury builds up, it's fat soluble, it stays in fats, and it builds up as far as not only bioaccumulation in the organism, but also how it relates to the food chain as well. So there's a number of things that depend on the toxicity of something, and there's a whole bunch of them for you to look at. There's also responses. The organism will have a response to the chemical in some way, shape, or form. One of those responses could be acute effects or acute response, and that's where the organism immediately reacts either to a rapidly harmful reaction. So it's an immediate reaction. It's an acute effect. It happens right then and there after the person is given the dosage or the chemical. The next type is a chronic effect or a chronic response, and that can be permanent, a long-lasting harmful effect. So the first one, acute, can be like immediate, like you get, for instance, more biological because this has to do with bacteria. Let's say you get food poisoning. Immediately you respond to that by throwing up, but in a few hours it's gone. You're rid of it. Your body gets rid of that. That's an acute effect. A chronic effect is where you could get little dosages of that, and it could last a long time and really build up over time. It can be permanently long-lasting harmful effects that can happen. For instance, like you might be able to give a dosage of radiation. Okay, for instance, from like Chernobyl. That had long-lasting effects on people, not necessarily immediate ones, but it could have had an immediate effect on them, but it also is a long-lasting effect that's going to go not even really affect them, but maybe also their offspring as well, if they were carrying at the time. So this is a science estimating human exposure to chemicals and measuring their effects. So you can see all these different things have to go into a scientific measuring model, usually done by computer. Metabolism, accumulation, excretion, the lungs, intestines, and absorption rates, the genetic predisposition, the personal habits, the lifestyle, the overall health, the nutritional things, the food and pesticide levels, soil, dust, water pollution, air pollution. All that goes into a model and predicts the level of toxicant in a person. Or toxicity in a person based on a specific chemical. Also, we've done case studies in protecting children because there was some analysis done using umbilical cord. So after the umbilical cord is cut, the baby is born, it's breathing fine, it's getting all the nutrition needs now on its own without the umbilical cord. But the umbilical cord was tested as far as running toxicological reports on them and trying to figure out what was that child already predisposed to these chemicals based on what the mother was eating and giving to her child. And infants and children are the most susceptible to toxic effects of chemicals because they eat, they drink water, and breathe more per unit of body weight than adults. They're growing. They need lots of stuff. They're intaking lots in order to grow. And so that's why they are more susceptible. They're also because they put their fingers in their mouth all the time and their toes and everything else. And all sorts of objects they can fit in their mouth, and that also carries dangerous things with it. And they're also less well-developed immune systems. Remember, they're still relying on that past immunity from their mother in the early stages, which isn't very strong until they get their active immunity, and that isn't really well developed yet in order to kind of ward off some of those dangerous chemicals or things that they're being exposed to. We also learned about dose response curve, or you will be looking at the LD50 and the median lethal dosage, which basically LD50 means killing 50% of the tested subjects. They don't have to be animals, I guess, whatever test subject you choose. There are two types of response models. There's a non-threshold dose response model, which basically means as you increase your dosage, the more deaths you see. A threshold dose response model is you might be able to give a little bit, and kind of increase that, and you might see no effect at all until it reaches a certain threshold or a level of dosage, and then you start seeing negative response from the population that you're testing on. So that is what we call the difference between non-threshold and threshold. I think we'll look at a graph of this a little bit later. One of the things is that a lot of times we test on living creatures like animals, crickets, rats, mice, bunnies, everything else. Can that be perfectly extrapolated to humans? Not necessarily, but there's some data models and some predicting models that can be utilized. There are more humane methods that rather than using animals, we can replace animals with other models like computer simulations, tissue culturing, individual animal cells, taking bits of skin cells, not the whole animal, maybe even using chicken eggs and membrane, if the eggs are very thin, to see how that will react to the embryo. So there's a couple other things that are more humane rather than using live, fully adult animals. This is what the lethal dose or LD50 curve goes. I think in our lab we made a mistake, that's okay. And looked at, we put the dosage up on the y-axis, on the x-axis we put the population. It looks better if you do it with the dose on the bottom, which is the x-axis, and then your percent kill, which is your dependent variable on your y-axis. So, but we got the same result pretty much. We still figured out the exact same what the lethal dosage was, but that's usually how you would graph that. So think about that if you ever take that AP exam. Okay. Where it killed 50% of the population, so this dose would be 7 roughly hypothetical units. We don't even know what the unit is, milliliters, grams, whatever, of the toxin that would kill 50% of the population. All right. Toxicity ratings and average lethal dosage for humans. You can see super toxic will be less than 5 milligrams per kilogram of body weight. That's super toxic, less than 7 drops. Examples would be like nerve gas, mushroom toxin, botulism toxin. Some pretty extreme examples. I should say super. Extreme toxic would be like cyanide, heroin, nicotine. That's 7 drops to 1 teaspoon, 5 to 50 milligrams per kilogram of body weight. Very toxic would be like morphine, codeine, mercury salts, things like that, 50 to 500 milligrams per kilogram of body weight. And then again, all the way down to essentially non-toxic like water, table sugar. You have to have 15,000 milligrams per kilogram of body weight to really have a lethal dosage of it. So that's only where it kills 50% of the population. So that isn't. That's just giving you a look at some different types of substances that could have some impacts. This is the different types of threshold versus non-threshold. Again, no threshold is when if you start as you increase dosage, you will see some sort of response. This is a linear response where it actually increases. It might still be a slow increase, not be exact. A nice line going through there. But again, it has some sort of effect. It will increase slowly. This is a little bit more of an increase, a linear response or nonlinear, but it's still increasing as dosage is given. This one is a threshold response. It takes a little bit of, you have to have a little bit of dosage before. You can increase and have no response at all until you get to a certain threshold level, which is here, and then you start to see a response. Now, it could actually be a nonlinear and still go this way, but we're just showing that way. You're just seeing the difference between the threshold and non-threshold response. All right. There are other ways to estimate harmful effects of chemicals. One of them are epidemiological studies. Basically what happens with epidemiological studies is that they're taking a look at actual cases that have happened in the past and interviews and kind of talking more about people with that. And so that is one of those that says which compare health of people exposed to a particular chemical or experimental group with the health of a similar group of people that were not exposed. So the limitations of this is that we're not going to actively expose people to some sort of chemical or toxin because that's inhumane. We have to look at cases like, for instance, a natural disaster, well, not natural, but a human disaster like Chernobyl, and take a look and compare those people to a group of people that have not been affected by radiation. There are too few people tested because there's hopefully not a lot of those major disasters or groups of people being exposed that's actually happening. It also takes a lot of length of time. Can you link the results with other chemicals? And it can't really be used for new hazards because you're only focusing on that one single chemical or that one situation that was given. So we have, we're looking at case studies and epidemiological studies, but they're not as helpful for us if there's not as many cases going on. Also, trace toxins, trace toxic chemicals can be harmful. We don't know yet, but we know that there's trace levels of different toxic chemicals in lots of things. If they have bioaccumulate in our bodies, that can obviously be a bad thing. But we don't really know the dangers of them all. Some of them are not going to build up in our bodies, so are they still going to affect us some way later down the line? We just don't know. The tests they need to be more sensitive to figure this out. But on this page in your book, I think it's page 48, 458 in your book, talks a little bit more about all the different types of household items that have trace chemicals found in them that maybe could be potentially harmful down the line. I know some of you are using these for your public service announcement project. So just make sure they remember they're trace chemicals. A lot of these maybe to have an impact, but again, we don't know the long-term effects of trace chemicals. Why do we know so little about the harmful effects of chemicals? Well, there are severe limitations in estimating toxicity levels within people. And again, because we're not going to actively test it on people, it's very hard to get an exact knowledge of what's going to happen if we expose them to different amounts of this chemical. So there's a lot of variance there, which is kind of why it's hard to estimate specifically and get good results. The pollution prevention and precautionary principle are so key. It says those introducing a new chemical or new technology would have to follow strategies. In this case, we kind of, with chemicals and new products, we kind of they're kind of flipped backwards to the court of law, which is, and they're actually guilty until proven innocent. So a new product actually is considered harmful by the Food and Drug Administration Act, like when you're taking a new pill, until it actually can be proven safe. Which is why a lot of times they have those disclaimers saying this could cause this, if you try this drug. And existing chemicals and technology that appear to cause significant harm must be removed. So not only, I know sometimes you see a lot of those local lawyer commercials like, have you been exposed to this, or did you take this drug? We think you can seek lots of remnant pay and all that kind of stuff, because they weren't full, they were put on the market maybe, and with a lot of disclaimers, but caused major effects later that weren't anticipated. So a lot of times we need to consider those chemicals or those drugs or things guilty until proven innocent. All right. Oh, in 2000, there's another that little thing there. In the year 2000, there's a global treaty to ban or phase out all the dirty dozens, which are the POPs, and we'll talk about that. Those are persistent organic pollutants. This includes DDT and PCBs, toxins, and all sorts of things that were actually signed and trying to remove those and phase out those about a dozen different persistent organic pollutants. Individuals about Ray Turner and his refrigerator. We knew about CFCs in '74 and them being depleting the ozone layer. In '92, they had agreement to phase out CFCs and other ozone chemicals. The Montreal Protocol, as well as a couple other different treaties that were signed. But also, Ray Turner figured out that, they used a lot of CFCs in his company to clean circuit boards and clean things in computers. He decided to figure out and test other things, and one of them he found was a citrus-based solvent that actually helped to clean the circuit boards instead of using the CFCs, things that he was using previously. So that's just one little case study, individual matter. Ray Turner figuring out that, hey, he learned that CFCs were dangerous. He learned about the treaties that were signed, and so he started to figure out alternative methods instead of phasing out some of the CFCs that he was using in his own field. So that ends chapter 17.4. We'll see you guys next time for 17.5 on looking at how we perceive risks and how can we avoid the worst of them.